Hi, I'm Gelbart from Chemistry and Biochemistry. I want to take these few minutes to give you a feeling for how we work with viruses in my lab and why we're so deeply intrigued by them. I want to share some slides with you. And in particular, let's start by looking at what a gene looks like in double-stranded DNA form versus what it looks like in single-stranded RNA form. This is the DNA template from which we in vitro transcribe the mRNA for gene 3 of a 4-gene virus we work on. That's right, a virus can have as few as 4 genes. This is a plant virus, CCMV. And the point of this is to show, it's a cryo-electron micrograph, showing how much more compact single-stranded RNA is than double-stranded DNA. You have the same amount of genetic information. In fact, it's identical genetic information. Associated with this is the fact that the RNA gene as an RNA molecule is about the same size as the virus particle that's formed when you add the capsid protein for this virus to the RNA in physiological buffer solution. And this is in stark contrast to the situation with double-stranded DNA viruses, where the DNA genome requires a much bigger volume to be thermally equilibrated than the size of the capsid inside which it is packaged, but not spontaneously. Instead, a very strong motor protein, one of the viral gene products, has to do a lot of work to package the DNA into a preformed capsid. Whereas with RNA viruses, we can simply mix the RNA gene with the viral capsid protein in appropriate buffer solution. And when we analyze the reaction mix, we find when we're using the capsid protein from this plant virus CCMV, that we can package any RNA, doesn't have to be the viral RNA, it can be RNA corresponding to a particular gene of interest, a therapeutic gene, a reporter gene, a viral antigen for a vaccine. And we don't just package this gene, we first add it in mRNA form to the mRNA coding for an RNA-dependent RNA polymerase, an RNA replicase gene, which we get from any of several RNA virus genomes we work with. So the point is that this mRNA in a cell will first be strongly amplified because it's part of the mRNA that codes for the RNA replicase. And after strong amplification, it will be translated. So you get very strong protein expression. We package this self-amplifying form of the messenger RNA of interest into these perfectly monodisperse virus-like particles, as I just explained to you. And we get particles that are indistinguishable from the wild-type virus, highly monodisperse, thermally stable. They protect the RNA against nucleases. And yet, when they're in mammalian cells, even though the protein shell is formed from the capsid protein of a plant virus, the RNA content is made accessible to the ribosomal machinery of the host cell in the host cell cytoplasm. So you never need to involve the nucleus of the host cell in the amplification of this RNA and its translation to proteins of interest. We use this idea to study RNA replication, viral self-assembly, but also to work on many translational medicine projects. Currently, we're working on a COVID-19 vaccine platform that involves self-amplifying mRNA coding for T-cell epitopes of SARS-2. We in vitro package that RNA, and we can target the resulting VLPs to antigen-presenting cells. It's the basic idea there. If you're interested in that, you could join that project for a quarter or for longer if you're interested. We also work on packaging these RNAs in cylindrical virus-like particles using capsid protein from other plant viruses. And we look at the effect of 2D versus 3D cell culture to see how viruses grow and how RNA is replicated. Please check out our website, contact me by email, and look forward to meeting you soon, and wish you a very good start of the year. Good luck to you. All the best. Bye-bye now.